Welcome everybody. I'd like to welcome you to the first in a series of seminars hosted by the UCD Research Partners team that's going to be focused on COVID-19 research in UCD. The purpose of this seminar series is to showcase some of the great COVID-19 related research taking place at UCD across the 6 colleges, and also to help facilitate greater interdisciplinary collaboration within UCD and with any external partners as well. Just to say, like today, all future seminars in the series will take place weekly at 2 o'clock on a Wednesday, right up to mid-December. And the format of these sessions over up to 30 minutes will begin with hearing from our speaker or speakers before moving on to a short Q&A session from the audience. And at that point, I'd just like to say, anyone listening, if you'd like to submit any questions, please do so via the questions and answers section on your login. Today we have 2 guest speakers. Dr. Barry Kevane is a consultant hematologist at the Mater Hospital, and Professor Patricia Maguire, who is professor in UCD School of Biomolecular and Biomedical Science. Just to give you a little bit of bios on our 2 speakers. So Barry has completed his undergrad medical training in 2008. At UCC, following initial postgrad training in general internal medicine and hematology. He then undertook a period of full-time clinical and translational research in the UCD Conway Sphere Research Group, leading to the award of his PhD in translational medicine in 2017. In 2018, he was then appointed to a consultant post at the National Coagulation Centre in St. James's Hospital, and was subsequently appointed to his current post of consultant hematologist with specialist interest in venous thromboembolism at the Mater University Hospital and Ireland East Hospital Group. That was in September 2019. And Dr. Kevane is also a principal investigator with the UCD Conway Sphere Research Group, and his primary area of research interest is in the field of thrombosis and hemostasis. Patricia is the professor, as I said, in our UCD SPBS school. She is a principal investigator and fellow at the UCD Conway Institute, and she directs the Conway Sphere Group together with Professor Fanula Ni-Ainle and Dr. Barry Kevane, where they use unique tools to diagnose and understand a host of inflammatory-based diseases, including early onset preeclampsia, multiple sclerosis, venous thromboembolism, and now COVID-19. Patricia is also the director of the UCD Institute for Discovery, where their mission is to build and cultivate interdisciplinary connections across the UCD research community through initiatives around emerging areas and technologies, such as the recent event that they hosted last week on AI in healthcare. So today, our speakers are going to be talking about their research that focuses on characterizing the hemostatic or vascular derangements which arise in COVID-19, with a view to addressing urgent clinical dilemmas and developing novel solutions for existing diagnostic or therapeutic challenges. And at this point, I'd like to pass the microphone over to our 2 guests. And I think, Barry, you might be wanting to talk first. Over to you, Barry.
Thanks, Kevin. And I have to say, both Patricia and I are really delighted to have been invited to give a talk on the inaugural session of this research seminar series. We're delighted to be able to give you an overview of our cocoon study and to share some of our preliminary results that we generated to date. So I guess at this point, next slide please, none of us need to remind you all that COVID-19 is clearly a disease that is primarily characterized by inflammation affecting the lungs. So this disease is characterized clinically by fever, respiratory symptoms, and pneumonitis. One of the features which has been very consistent since the initial reports of this pandemic in China earlier in the year is that patients with COVID-19, particularly with severe COVID-19, have a very distinct and unusual pattern of coagulopathy characterized by the presence of elevated markers of blood coagulation, such as D-dimer, elevated fibrinogen, and a relative lack of overt evidence of disseminated vascular coagulation, or DVC, which is a coagulation condition which frequently complicates patients with severe inflammatory disorders such as severe pneumonitis. So this distinct coagulopathy, very much in keeping with a hypercoagulable state, in COVID-19. And what is probably even more intriguing, and what has since emerged, and again has been very consistent in data both from Asia but also from other parts of the world such as Europe and the United States, is that this pattern of a hypercoagulable phenotype and particularly high levels of D-dimer appear to correlate with the severity of inflammatory illness and indeed with risk of mortality. It's not surprising given the evidence of I suppose, elevated coagulation activation in COVID-19 that we were quite concerned about the potential risk of thrombosis affecting these patients. And indeed, it has been very, I suppose, consistently reported that rates of thrombotic complications appear to be much higher than would be expected in patients with disease of a similar severity. And what's even more worrying from a clinician's point of view is that these thrombotic complications are occurring despite treatment with anticoagulant therapy, and of course are potentially life-threatening. The majority of events, of course, today being pulmonary embolism. The etiology of thrombotic risk is really unclear in this setting. Certainly, there's been a huge drive internationally with increased, I suppose, in terms of studying the precise mechanism that COVID-19 induces a hypercoagulable state, but as of yet, the true etiology remains unclear. And as a consequence of this, our optimal preventative and treatment strategies have yet to be fully determined. Next slide, please. So the cocoon study is a multi-center clinical and translational study, which both myself and Patricia and Fionnuala have been developing over the past number of months. We're based primarily in the Mater Hospital, but we also have in the Kevane Institute, in the Kevane Sphere Group. We also have a collaboration initiated recently with the Infectious Diseases Cohort Study. And the purpose of this cocoon study will be to try to characterize some of the mechanisms underlying hypercoagulability in COVID-19 and also to potentially identify non-prognostic and diagnostic biomarkers which may be of use clinically. And we're delighted to have been among the first biomedical studies funded by the recent SFI COVID reimbursement. So with regards to, I guess, the workflow, the work packages that we intend to apply in this study, and next slide, please, Trish. So I should also mention actually that we have a huge number of collaborators both nationally and internationally, some of whom are mentioned here. So this is truly a multidisciplinary and a multi-center study. And next slide, please, Trish. So with regards to our workflow patterns, our workflow plan, briefly, I guess we will start off by undertaking a detailed characterization of patterns of hypercoagulability in plasma samples obtained from patients with COVID-19 and healthy controls as well as hospitalized non-COVID controls. This will include calibrated ultimate thrombography, which is a technique which we have established in our laboratory and which we have used in multiple studies in the past looking at different disease states. And it allows us to characterize in great detail the etiology of hypercoagulability in different disease states, and this will be applied to our COVID population. I think Trish will speak now to the some of the other techniques which we'll be using along the way as well.
Great, thanks, Barry. Can everybody hear me okay? Kevin, can you hear me? Yeah, great. Okay, so yeah, super. I'm just going to take over from Barry now and talk about the I suppose the different work packages that are involved in the cocoon study. So these are, I suppose, there's a number of different work packages involved. We're really interested in the role of platelets in COVID-19. I don't know, Kevin, can you see my arrow as I use?
Yeah.
Yeah, super, that's great, just double checking. So we're really interested in the role of platelets, I suppose, you know, from a clotting perspective. Platelets are involved, and also the role of the hemostasis as well. So we're looking on it from both aspects of clotting. So we're really interested in the role of platelets, and we have a number of work packages involved in that. I'm going to talk to you today really about two that are, I suppose, the most developed of these. But if anybody's any interest in the other work package that we have, please do contact us. So I'm not going to talk about this particular one where we're actually doing some proteomic analysis of on the blood samples that we've collected, but it's just about to start, that work package. And we're also going to look at some cyclic nucleotide signaling. Sorry, that's not supposed to happen. We're also supposed to look at some cyclic nucleotide signaling and that is another work package that'll be kicking off soon. We're going to do some analysis of circulating extracellular vesicles in the plasma of these patients as well. So the samples are banked and ready to go. It's just a matter of accessing them and running these on by mass spectrometry. And then, as Barry talked about, we're going to do some thrombin generation assays. So that work is almost complete, and looking basically at how fast thrombin is generated in the bloods of these patients. And it looks like, from our early results, it looks like it's a lot faster. So thrombin generation actually happens much quicker in these patients. So the two, I suppose, that I'm going to go through in a little bit more detail today are these two particular ones. So, oh, sorry, apologies. So the first one is looking at a hyperactive phenotype that we found in circulating platelets. So I'm going to talk a little bit more about this, and then we're also going to talk about, and I don't know why my slides keep doing that, apologies, always when you're live things go wrong, but so we're also going to look at some algorithms that we've been developing on the clinical data that we've collected. So I'm going to talk about these two in a bit more detail. So first, I'm going to talk a little bit about the hyperactive phenotype in circulating platelets. So we are not, I suppose, the only lab that has done this globally. There's a group in Canada and a group in the US who've also reported a hyperactive phenotype in platelets in COVID-19. But what we've done is slightly different. So we've actually taken, and this was Barry Fanula and the amazing team in the Mater Hospital have collected over 54 patients with COVID-19. So, and we can split these into based on the severity of their disease. So we have a number of patients who have, I suppose, what's classified as non-severe COVID-19. And then the people who are classified as severe COVID-19 are those who require intensive care or are currently in ICU when we took their blood sample. And the interesting thing is that we were able to match these against hospitalized controls. So all our data here has been matched against these hospitalized controls. So who are these? These are people that are in hospital but haven't required to be tested for a COVID, haven't showed any symptoms, I should say, of COVID-19. So we were able then to show that, so we tested these patients, took blood on day 0, but then also took blood either if when they were transferred to ICU, or for those who weren't transferred to ICU 7 days post-admission. So we were able to look at certain parameters in their blood, and this particular parameter that we've highlighted here is mean platelet volume, and basically it's almost akin to a measure of the size of the platelets that are made by the megakaryocytes in the bone marrow. So what was really interesting is that we could separate out severity of the COVID-19 infection based on their counts even at day 0. So what do I mean by this? Even when they arrived in the hospital, we could separate out those who had severe COVID from non-severe. So the other thing we looked at as well is we did some platelet activity assays. And this is some particularly, I suppose, we did a lot of different analysis, but this is one, I suppose, that I just wanted to highlight because it's particularly profound data that I suppose we've never seen before in any other disorder. And it's looking at, so platelets have these preformed granules inside them, and a particular granule called dense granules that are preformed inside the platelet. They have a lot of different small molecules inside them, and when platelets are activated, they actually basically release out the contents of these granules into the external environment. And one of those small molecules that they have stored in there is ADP. So this is a very profound, this can be a profound agonist. It basically acts back on the platelets and drives the platelets to clot. So what we saw, this for us was a really interesting result. What we saw is we saw almost, so in the non-severe COVID patients, we saw 30 times more ADP being able to be secreted out of these from these granules. So what do I mean by this? I mean that, so if you just put a small dose of agonist on regular human healthy platelets, you get an amount of ADP being released. And when we did these with the hospitalized, so the regular hospitalized controls, we saw an amount of ADP being released, and that's what's shown in these two here. But you can see immediately, and if you just note the y-axis here, you can see immediately that much more, when we put the same dose of agonist on these platelets, we just saw a massive amount of ADP being released. And this was 90-fold higher in some of the severe COVIDs. So just a quick flavor of the data we've been managed to produce already. But just in total, what we saw is, and this, if anybody's really interested in all the data, because I just wanted to keep the presentation to 10 minutes today, but if anybody's interested in this, we've got this up on Med Archives, and this is currently under review, this manuscript at the moment, and the manuscript is entitled COVID-19 Induces a Hyperactive Phenotype in Circulating Platelets. And the main things we found is that our routine blood test can stratify the patient risk or the severity of the disease, even on admission to the hospital. We also, and I just showed you this data a second ago, they have COVID-19 platelets have a decreased activation threshold, which means when you put even a tiny amount of agonist on them, they basically just, boom, release all of their contents. Then, so we also then, in our conclusion really was that the COVID-19 infection seems to induce a really hyperactive and really hypersensitive platelet. And we did a lot more analysis. We looked for a lot of different, we did a lot of different platelet assays. And if you're interested, as I said, you can have a look at the full paper up on Med Archives. So the other data that I wanted to highlight today was a risk stratification algorithm that we've been doing for COVID-19. So again, this is down to the incredible clinical data that Barry, Fanula and the team have collected. And so we were lucky enough to be able to work with the AI for Healthcare Hub in UCD, which just got launched last week. So that they're working with SAS Institute, who basically are data analytics specialists. So what we were able to do was we were able to take all of our data, all of our patient data, and look for patterns in the data. So we use, so this is very basic statistical analysis, decision tree analysis, but we were able to again separate out. So this is just a confusion matrix, and we were able to separate out and predict who would become severe over time. So if anybody's interested in that, again, you can contact me by email, and I can go through this in more detail, or ask some questions after this presentation. So that's the overview. Just, there's a lot of people involved here. I just noticed, Barry, that we're missing you from the picture, so apologies. But there's a lot of people, you know, involved in this to make this happen, and we are absolutely delighted to receive SFI funding from this. So Barry, I'm going to hand back to you.
Okay, I think there was an issue with audio there.
Yeah, I think you're quite echoey, Barry.
Is that any better? Sorry.
Yeah, much better. Yeah, there you go.
Sorry.
Yeah, sounds good.
Thanks, Trish, for really summarising the work. I guess the one that we just wanted to finish off with was just to maybe highlight that Later this week on Friday, Fanuda and myself and most Breslin, who are all matter consultants, we are hosting our annual BCE Dublin meetings. This is a conference that's been run annually for the last 5 years. We've been really lucky over the last few years to attract really high quality international speakers, and that's the case again this year. The good thing this year, however, is that it's all virtual, and so registration is free. And there are still a few slots that are left for potential if people want to attend. And if there are any clinicians on the line, the RCPI have also assigned 6 CPD points, which may be of interest to clinicians who may be listening. So this year we have a really good lineup again, a lot of international speakers, such as Professor Hunt from London, who's an expert in thrombosis and speaking particularly with respect to immunothrombosis and COVID. And we also have Professor Sosky from Harvard and Massachusetts General Hospital, who will also speak about incidents of bleeding and thrombosis from their experience in Boston. So a really good lineup, and I would certainly encourage anyone who might be interested to register and to attend virtually on the day. Thanks. So I'm sure if there are questions, Kevin, maybe.
Thanks very much. Yeah. I'd like to ask a question myself, actually, before I go to the audience. Barry, for yourself, actually, and just say that it's still a bit crackly on your end, but I think it is better than it was earlier, and just in case you're wondering. But the question I wanted to put to you was, in terms of the coagulation abnormalities that are seen in COVID-19, is it something that differs than what would be seen in other kind of acute infective or inflammatory illnesses like the flu or?
Yeah, and that's, I suppose, the real hub or the real core of the issue is that we always see coagulation abnormalities in patients who are very unwell with inflammatory illnesses. But what we're seeing with COVID-19 is that they are really, I suppose, excessively hypercoagulable than what we would normally expect. And from the data we've seen reported, particularly from autopsy studies, we're seeing evidence of widespread microvascular thrombosis, particularly in the lungs, and that's probably contributing to respiratory failure and death in these patients, even in these patients who may not necessarily have had an overt clinical diagnosis of thrombosis maybe prior to death. So it's likely that coagulation activation in COVID-19 is different and unique and specific to this particular virus in contrast to other conditions. And that's why, treating these patients is becoming increasingly complicated for us, because not only are they developing blood clots, but even, say, a lot of these patients in the intensive care unit are requiring dialysis or special forms of ventilatory supports with instruments such as ECMO, which are prone to clotting at the best of times, but even within the COVID-19 population, they're clotting a lot more than they normally would. And particularly from the point of ECMO, which is a form of respiratory or any stage of respiratory support, a clot in an ECMO circuit is immediately life-threatening. So with regards to, I suppose, that question, absolutely there are differences in hypercoagulable phenotype in COVID-19, which are unique to COVID-19. And if we can, I suppose, develop a strategy towards targeting this through translational research and also clinical research, I think would be huge benefit.
Yeah. I guess to follow on from that then would be, how COVID patients are managed. I mean, is that something that you've seen change over the last 6 months or so from the clinical experience?
I think there's definitely a move internationally towards escalating anticoagulant therapy, even in scenarios where patients are being diagnosed because of the recognition of increased coagulation activation in these patients. Now, there are some clinical trials which are ongoing to evaluate this approach, and my colleague from Dublin is going to be leading the Irish, I suppose, the international study of coag, which would be assessing different doses of heparin in patients with COVID based on certain specific clinical criteria. So certainly there is a move internationally towards escalating therapy, but at the moment has been done without real evidence, but hopefully we will have more clinical trials prepared over the next few months that might help to find the appropriate dosing strategies. But a huge area that's really challenging for us.
Yeah, very interesting. Patricia, I was going to ask, in terms of the research that started all this, I mean, what made you look at the platelets in the first place in terms of COVID-19?
Yeah, I suppose, sorry, I just sounded a bit echoey there. Is that better?
Yeah, you sound good.
Yeah, sorry. Yeah, we've been working on platelets for a number of years, but we've also been working on the coagulation pathway as well. So, and that's, I suppose, what we do in Conway Sphere is we merge both sides of obviously the platelets, the clotting aspect from the platelet side, and also then from the coagulation pathway, because there's numerous interactions. And we're also interested in extracellular vesicles, circulating extracellular vesicles, because I suppose 70% of all circulating extracellular vesicles are derived from platelets. So it's just something we've been interested in. When we saw especially what Barry had just been speaking about, the papers, the post-mortem papers, yeah, that really, you know, there was clotting right through these poor people. So I think, you know, from a scientific perspective, I think we had to look at the platelet, and we were very lucky that we could access in the samples.
Okay, and I saw one of your other work packages talk about platelet releasate.
Yeah.
Can you explain exactly what that is?
Yeah, so actually what I described during the presentation, when the platelets are activated and they basically secrete out their contents of preformed granules. So I spoke about dense granules, but platelets actually have another 2 types of granules as well. And we've actually been looking at the granular contents of platelets, and we coined the term platelet releasate back in 2004. So we look at these, we do omics analysis on that platelet releasate. We've extensively done it, not just in COVID, or well, we will do it in COVID, but we've done it in it for a number of different inflammatory type diseases over the last number of years. Okay,
great. So you can apply it now to COVID, so that'll be
it's been basically it's the differential changes have been very interesting. It's led to a number of very interesting diagnostics in different diseases, in specifically preeclampsia in pregnant women, and also for different types of multiple sclerosis. That allows us to separate out the different types. So we thought it'd be really good to look at the COVID patients. So we're hoping, we've just processing the samples now, and we're hoping to get them onto the mass spectrometer in the Conway Institute, I suppose, sometime in the next month. So the idea is that we'll be able to find new diagnostics, hopefully, or will at least be able to give us an understanding of how the contents of this platelet releasate is changing, because it's really important. I suppose pathophysiologically, it's really important, because there's huge amounts of effectors in there, and the changes in those effectors, in the amounts of those effectors, even might have a profound change on how those platelets activate.
Very good, very good. We're kind of coming up close to our end time, so I might get one last question in. And in terms of the entire study, I mean, I guess it's maybe a clinical question for Barry. What the kind of potential clinical, implications are for the translational work that you guys are doing in the study? Can you envision what it might be? All things going well?
Yeah, I guess from I suppose the urgent clinical needs really are to optimize our preventative and treatment strategies for patients who are developing strange and unusual patterns of widespread microvascular thrombosis. We know, based on the current our current approach of sort of standard dosing of antibiotics doesn't appear to be effective. So I guess what we would hope is that the translational work we're undertaking would potentially point to our general hypotheses that could be further explored in clinical research. So certainly the paperwork which Patricia has described there is something which is really interesting, and currently, looking at the potential of the role of antiplatelet therapy, for example, in these patients, is something which we could consider or could form the hypothesis of a trial based on the work we were doing on the road. But I guess the point of the study, or the reason why we all decided to develop this work, was to try and I suppose investigate or try to target these very significant unmet clinical needs. And currently the big one is optimizing our prevention strategies. You're muted there, Kevane.
Thank you very much. So I think we're coming up on the time, which is less than a minute. So I think I'd like to end our event there today and to thank our speakers, Patricia and Barry, for taking the time to speak with us today and sharing with us your very interesting research, and also to the audience for their questions. And if you'd like to continue the conversation or submit any further questions on the work that our speakers are doing, could you please go to our COVID-19 Basecamp team that you would have signed up to upon registration for the event. And just say then, next week we'll be having another session in our series, the second one, that will be with Dr. Klaas Kirchel with his research topic, History in COVID-19, Untangling the Roots, Trajectories and Interpretations of a Pandemic. Okay, thank you very much, everybody, and thanks very much again to our speakers. Thanks, Patricia.
Thank you. Bye.